How is amelogenin used in forensics? Have you ever wondered how forensic scientists determine the biological sex of an individual from DNA evidence? One of the key tools they use is called amelogenin testing. This method is essential in true crime investigations, especially when identifying unknown human remains or biological samples from crime scenes. Amelogenin is a gene located on both the X and Y chromosomes, but there are slight differences in size between the two versions. This unique characteristic allows forensic experts to tell whether a sample comes from a male or female. If the sample contains only the X chromosome version, known as amelox, it indicates a female. If both the X and Y chromosome versions, amelox and amely, are present, it indicates a male. In many forensic investigations, determining the sex of a sample is one of the first steps. Amelogenin testing is often integrated into genetic profiling systems because it provides a quick and reliable way to establish whether the DNA sample is male or female. This information can significantly narrow down the list of potential suspects or victims, guiding further investigative efforts. The process begins with extracting DNA from biological materials like blood, saliva, or even dental pulp. Forensic scientists then use a technique called polymerase chain reaction to amplify the amelogenin gene regions. The amplified DNA fragments differ in size depending on whether they come from the X or Y chromosome. This size difference allows laboratory analysts to easily distinguish between male and female samples. Amelogenin testing is particularly useful in forensic odontology, where DNA extracted from teeth can be analyzed. Teeth are excellent at protecting DNA, even in harsh conditions. This makes amelogenin testing effective in cases involving severely decomposed or burned remains, where other tissues may be degraded or absent. However, there are some limitations to relying solely on amelogenin for sex determination. Certain individuals may have deletions or mutations in the Y chromosome amelogenin gene. This can lead to false results, showing a female result in males if only this marker is tested. To avoid misidentification, forensic protocols often include additional Y chromosome markers, such as the SRY gene or Y-short tandem repeat markers, to confirm male sex in ambiguous cases. In summary, amelogenin testing is a fundamental tool in forensic science for determining biological sex. It helps establish whether biological evidence is male or female, aiding in human identification and criminal investigations. While it is highly useful, it is often complemented by other genetic markers to ensure accuracy and avoid errors in forensic casework.